So let's move now from working buffer problems to actually considering buffers that are a little bit more biological in nature. The one that I've described so far has been simple buffers. Simple buffers have a single proton that can come on or go off. So acetic acid, for example, we saw had HAC, the AC being the A part, going to H plus AC minus. That is only one proton that can come off. When we look in cells, cells are full of things like amino acids, for example, that have multiple protons that can come off of one molecule. And those multiple protons that can come off, in each case, is coming from a weak acid. Okay? So these types of systems are more complicated than we see for a simple system. So let's consider, first of all, acetic acid, which was a simple system, which had a titration plot that looked like this. We saw a rapid increase in the pH until we got into a buffering range, flattening of the curve, and then a rise as we got outside that buffering range. The maximum buffering capacity occurred at the pKa value. Okay? Now, maximum buffering occurs when salt equals acid, and that's what's happening right here. You'll notice that this buffer doesn't resist change in pH over the entire range. In this range, we see very rapid increases in pH with just tiny amounts of hydroxide being added, where here we see a large change in hydroxide being added, but very small changes in the pH value. And then again over here, we see small amounts of hydroxide added and a large change in pH. So in this region, from about here to about here, we have the buffering region, with the maximum of it being the center right there. A good rule of thumb for any buffer is that a buffer acts as a buffer within a pH range +1 or -1 from the pKa value. So if this were acetic acid, for example, acetic acid has a pKa value of 3.76, we would expect it would act as a buffer from about 3.76 over here up to about 5.76 over here. If we were in pHs outside of that range, acetic acid would not act like a buffer, and we would see very rapid changes in pH as we change protons and hydroxide concentrations. Well, that's a simple weak acid. What if we have a more complicated weak acid? Well, a more complicated weak acid might be aspartic acid. Aspartic acid, to give you an idea of its structure, looks like this. Acetic acid, I will remind you, looks like this. Now, first of all, we can see quite a bit of difference. This only has 2 carbons, this has 4 carbons, but more importantly, this has 1 carboxyl group. This is the only part of the molecule that can ionize, that's right there. If it ionizes, that is, it loses its proton, we see that it becomes COO-, and so we have 2 choices, COOH, which is the acid, COO-, which is the salt. On the other hand, aspartic acid has 3 groups that can ionize. Each of the carboxyls that you can see here, and this amine can also ionize. Okay? Well, if it ionizes, what it actually does is it gains a proton, becomes NH3+. So it can exist as NH3+ or as NH2-. Again, this is from your basic biochemistry amino acid structures. This carboxyl can exist with a proton on or with a proton off, and this carboxyl can exist with the proton on or the proton off. So that means that this molecule of aspartic acid has 3 different groups that can ionize, and the question is, do they have 1 pKa value or do they have 3 pKa values? Well, it turns out that each of these ionizing groups has its own unique pKa value. Now, I'm going to show you in a second a titration curve that uses information about each of these values to predict what molecule will be present at any given pH. But suffice it to say that if we think of this guy as having 3 pKa values, we could actually predict what the titration curve would look like. It would look something like this. We would see a rapid increase until we got to the first pKa value where we would see flattening. We would see a rapid increase until we got to the second pKa value where we would see flattening. And we would see a rapid increase until we got to the third pKa value where we would see flattening. So that we would have pKa1, pKa2, and last we would have pKa3. So that's what we're going to see in the next slide I'm going to show you. Before I talk about the titration curve for aspartic acid, I want to work with this simple plot that I drew earlier for acetic acid and point out some things for you, because I think it'll be easier to understand here when we go to the more complex curve. Acetic acid, we remember, has the following structure: COOH, CH3. Okay? If the proton is lost, we end up with a structure that looks like this. So this is the salt, and this is the acid, because the acid has one more proton than the salt does. If we look at this buffering curve, we should remember a couple of things. First of all, when we have maximum buffering capacity, we have salt equals acid. So we would expect at this point, at this pH, we would have equal amounts of these two. And in fact, that's exactly what we have. We have equal amounts of the acid, this guy, and the salt, this guy. If we start putting more protons into the solution, we remember what happened. Well, from the Henderson-Hasselbalch equation, we said pH equals pKa plus the log salt over acid. And we remember that HA goes to H plus A minus. If we put protons in, we're going to favor this production. And if we take protons out, we're going to favor this guy right here. Okay? Now, if we have equal amounts, of course, this is 0. This log term becomes 0, and pH equals pKa. If we have more salt than acid, we'll have a higher pH. If we have more acid than salt, we'll have a lower pH, and that's consistent with what I'm showing you down here. Well, why do I show you all that? I want to show you that because I want you to think about what molecules might be present. In this range right here, the buffering range, we've got a mix of S and A. A little bit more A over here, a little bit more S over here, but not too wide of a ranging concentration of each. However, if we get outside of that buffering region, more than one pH unit away from the pKa value, we can say mostly what's there. Mostly what's there is acid. At this low pH value, we see mostly acid. At the high pH value, we see mostly salt. Okay? So the salt that we're going to see is going to be here, and the acid that we're going to see, I should have drawn this a little differently, is right here. Okay? Now, that's important, okay, because as we're going to see from the aspartic acid, we can actually predict which molecule is present at each of the pH values that are there. But I want to emphasize something very important here. In this range, we don't say which one we have because we don't predominantly have one or the other. In fact, what we have in this range is a mix of the two. So we can only say we have predominantly one or the other when we're outside of that buffering range. At pHs above the buffering range, we're going to have primarily salt. At pHs below the buffering range, we're going to have primarily acid. Okay, now let's take a look at that long-promised titration curve that's precise, showing molecules for aspartic acid. It's shown right here. And like we predicted, it has three regions where we have flattening that correspond to buffering, and those three flattening regions correspond to the individual pKa values for each of the ionizable groups. When we look at aspartic acid, it's important to remember something over here, okay? So aspartic acid, I'm going to put its structure up over here first to start, and it looks like this. Okay, so that's the molecule that we're working with. And in fact, as I said, we have 3 ionizable groups. They can have the protons on or the protons off. And to start simply, I'm going to put all of the protons on. So we've got NH3. When I have a 3 there, that's a plus. I have a carboxyl group right here, and I have a carboxyl group right here. This carboxyl group is the one I call the alpha carboxyl group. This amine is what I call the alpha amine. And this carboxyl group is what I call the R group carboxyl group. And so I'll be referring to those as I go through the titration curve. Now, it turns out that, as I said, each of these individual groups has its own pKa values. So for this plot, we see the pKa values are as follows. For the alpha carboxyl, it's a value of 2.1. For the alpha amine, it's a value of 9.82. And for the R group carboxyl, it's a value of 3.86. So using that information, that's actually, actually having done the titration curve, one can see a titration curve that's very much like what you see on the screen right here. All right, so we have our 3 flattening regions. Here, for example, is the first flattening region that corresponds to pKa value 1, which is a value of 2.1, and it relates to the ionization here. The second pKa value corresponds to the ionization of this carboxyl ion right here, and it corresponds to a pKa value of 3.86. And the third ionization occurs way up here. It relates to the ionization of the amine group that you can see right here, and it has a value of 9.82. Now, using the information that I gave you earlier for acetic acid, how if we're outside of the buffering region, then we can predict which molecules are present. We can do that for a complicated molecule like aspartic acid. So let's do that. Let's start way down here, okay, where we've got a pH value that's, let's say, about 0, okay? Well, a value of 0 is way outside of the pH, of the pKa range of any of these groups. It's more than 1 pH unit away. And that means, therefore, that we will have all of these protons on. That is, we're going to have everything in the acid form, because when we are below, the pH is below the pKa value and it's outside of that pKa range of plus or minus 1, then we have the acid form. And the acid form, of course, has the proton on. So this guy, which had a pKa value of 2.1, if we're looking at it at pH 0, it's going to predominantly be in this form here with the proton on. Well, a value of 2.1 is obviously a long ways below this pKa value, meaning that this proton is also on, and a value of 0 is also a long ways away from this pKa value, meaning that this proton is on. So the structure that I've drawn here on the screen, as you can see up over here, corresponds to all of the protons being on. When I get into this region that you can see right here, where I have pKa value 1, I can't say what I've got present. I know that this proton will be on because a pH of 2.1 is more than 1 unit away from this pKa value, so this proton will be on. I know that this proton will be on because a pH of 2.1 is more than a pH unit away from this pKa value, but this one I can't say. Half of them are going to have the proton on, and half of them are going to have the proton off. So in the place where we have pKa values, we don't try to predict which molecule is present because we know we always will have a mix of molecules. So we only predict structures that correspond to structures outside of buffering ranges. So we can see here that this first molecule is present way down here. When we get outside of that first pH range, we lose the first proton. And losing the first proton, which will be the first proton lost? Well, the first proton lost is going to be this one, because remember at 2.1 we had half of them with the proton off and half of them with the proton on. And what we're going to see is something like this, where now we have a molecule that looks like this. Now, I recognize that in this range right here, okay, when I look in this range right here, it's 2.1 to 3.86 isn't 2 pH units apart, so we can only sort of wave our hand at that. But we can basically say for estimating purposes that this proton is off and that this proton is basically on because this value that you see right here is somewhat below the 3.86. So we can predict that we will see this molecule here, which corresponds to this one, which has this proton off, this proton on, and this proton on. When we get to that second pKa value, we're going to have a mix of something. And the mix that we're going to have is we're going to have half of the molecules with this proton on, half of the molecules with this proton off. What about these two? Well, at a value of 3.86, we're going to be above the pKa value for this one, so this proton will remain off. At a value of 3.86, we're more than a pH unit below this pKa, so this proton's going to remain on. Okay? And we're going to have half of the molecules here with a proton on, and half of the molecules with a proton off. Okay? So again, in that pK range, we don't say which one we have because we know we've got a mix of molecules. However, we get outside that pH range, as we would see over here. What do we see? Well, we would see exactly this right here, because now we're more than a pH unit above the pKa values of this one and of this one, meaning both of these protons will be off. On the other hand, this value here, which corresponds to roughly a pH of about 5 or 6, is more than a pH unit below this one, so therefore this proton will remain on. So at this point in the graph, this is what we will see. Well, we get up to the next pKa range, we have the next proton that starts to come off, and the only proton now that can come off is this guy right here. We're going to see half of the molecules look exactly like this with the proton on, and we're going to see half of the molecules that have lost that proton. So we will have something like this. Again, at that pKa value, we don't try to say that we've got one or the other because we know we've got a mix of those molecules. However, when we get outside of that pH range, as you can see up over here, we're going to completely lose that proton, and up over here, we can predict that we will have completely this molecule right here. Now, for estimating whether a proton is on or off of a given weak acid, we can use that rule that I've been illustrating for you here. And I'm going to state it for you here. And this is a rule that you'll find very helpful for estimating charge, as we're going to see in the next thing that we're going to do. Now, the rule is a rule that the students will need to know. I give you all kinds of equations on exams and all the equations that you need to know, but I don't give you this rule. So I want you to remember this rule, and I find that students get confused with this rule because they don't pay attention to the details of it. So here it goes. If you have a buffer system that has a pH that's 1 or more units below the pKa value, the proton is on and you have predominantly the weak acid. If you have a buffer system and the pH is 1 or more units above the pKa value, you may assume that the proton is off and you have predominantly the salt. If you learn those two rules the way I've told you, you'll be fine. But if you go in and you say, this is higher, proton is on or proton is off, you don't know which is higher, the pH or pKa. You will get confused and you will lose that. So we'll see that in the next problem I'm going to work. In the next problem, we're going to take the various things that we've learned about ionization of weak acids, specifically amino acids, and apply them to a solution where we have a polypeptide, and we predict the charge of that polypeptide based on the pKa values that are relevant and for the pH of the solution in which it is found. In this particular problem, we have a molecule that's shown here. It is arginine, tyrosine, serine, lysine, and aspartic acid. And I've told you that you can assume the following pKa values for this polypeptide. You may assume that the alpha amino group has a pKa value of 9.2. The alpha carboxyl group has a value of 2.2. The R group amine has a value of 11. And the R group carboxyl has a value of 4.4. And last, the sulfhydryl has a value of 8.0. So whenever you go to work through predicting charge of a polypeptide, you need to do things pretty much in the steps that I'm going to show you. Okay, so here is the sequence of that polypeptide. So we had arginine, we had tyrosine, serine, lysine, and aspartic acid. Now, I don't expect that you're going to memorize those abbreviations, and on an exam I would give you the full names, but I'm just abbreviating here so as to save space. Now, what you will need to know on an exam are the following things. You don't need to memorize structures, but as I said in the lectures, you need to know which amino acids ionize and how they ionize. Okay? Well, what can ionize? Alpha groups amines, alpha group carboxyls, and R groups. R groups can have amines, R groups can have carboxyls, and R groups can have sulfhydryls. All right. Now, if we look at this, when we go to predict the go to predict the charge of a polypeptide, we need to figure out all the things that are there. Well, fortunately, as I've mentioned in the class lectures, when we create peptide bonds between individual amino acids, we simplify the problem immensely. And the way we simplify it is the fact that the alpha carboxyl here is joined to an alpha amine, and neither one of those can then ionize. Neither one can ionize here, neither one can ionize here, and neither one can ionize here. So if you look at an individual polypeptide, it's only going to have a single alpha amine. And for the moment, I'm going to write it as an NH3 to put the proton on. So I'm going to call this the alpha amine. And by the way, in general, the convention is that we put the alpha amine of a polypeptide on the left, and we put the alpha carboxyl on the right. Okay? So any polypeptide is always going to have one alpha amine and one alpha carboxyl. Okay? If we then have that information, the only other thing we have to know to calculate the charge of this polypeptide is which of the R groups can ionize. And that's something that you need to know. Okay? You need to know that the amine-containing amino acids, arginine, lysine, and histidine, can all gain a proton and gain a +1 charge, or lose a proton and have a 0 charge. That is, the acid has a +1 charge, the salt has a 0 charge. You should know that cysteine has a sulfhydryl group, that is an SH. If the H is on, you have the acid and it has a 0 charge. If you have the proton off, you have the salt and it has a -1 charge. The other three that ionize for our purposes are, the other two that ionize are aspartic acid and glutamic acid. They each have an R group carboxyl, and those R group carboxyls have the proton on. They have a COOH with a charge of 0, that's the acid form, and they have a COO minus, they've lost the proton, have a minus charge when they're in the salt form. Well, now we look at this and we say, well, which of these amino acids then have an R group that can ionize? That's all we need to know. Alpha amine, alpha carboxyl, and R group. Well, this has an R group amine. Okay, and I'm just going to write it as an NH3 for the moment. Okay. Tyrosine does not have an R group that can ionize. No ion. Serine does not have an R group that can ionize. Okay. Lysine does have an R group that can ionize, and it's an R group amine. I'm going to write it as an NH3. By the way, these are pluses. Okay. Last, aspartic acid has an R group that can ionize, and I'm going to write it for the moment as a COOH because it's a carboxyl. Well, which one do I have? It really depends upon what the values are of the individual pKa values. Well, if we go back and we remind ourselves of those pKa values, we can see them right here. Alpha amine 9.2, alpha carboxyl 2.2, R group amine 11, R group carboxyl 4.4. We don't have any SHs in here. We don't have any that comes from cysteine. We don't have any cysteines in here, so we don't have to worry about that at all. Well, let's take that information then and say that we've got a mixture that is at a pH value of 1. What is the charge on this overall polypeptide? Well, we simply apply the rule that we learned earlier about the pH relevant to the pKa values of each of the ionizing groups. So for example, at a pH value of 1, 1 is more than 1 pH unit. Let's get the individual pKa values on here. So this was 9.2. This value was 11. This value was 11. This value here was 4.4. And this value over here was 2.2. All right? So if we apply our rule, we've got a pH of 1, and we go through these pKa values, we can decide if the proton is on or the proton is off. All right, well, pH of 1 is more than 1 unit below this pKa value here. That means the proton will be on, because when the pH is 1 or more units below the pKa value, we have the proton on, we have the acid form. This proton is going to be on. When that's the case, we have NH3, and we have a plus charge. If we go next over here, pH of 1, more than 1 unit below the pKa value, this proton is on, we have a plus charge. Same thing here, which also has a pKa value of 11. pH 1 is more than 1 unit below the pKa value of this guy, so we have the COOH. Proton is on, charge is 0. Okay? And last, we have this guy over here. pH is more than 1 unit below this pKa value. The proton is on. And so exactly as I have it drawn, that's the structure we would have at pH 1. What's the overall charge? Well, we've got plus 1, plus 1, plus 1, plus 0, plus 0. We have an overall charge of plus, let's write it down here. Charge equals plus 3. Okay. Well, what if we took that molecule and we said, well, we're going to have a pH of 3.3. What would be the charge then? Okay. Well, what would that be? Well, let's apply our rule again. 3.3 is more than 1 unit below this guy. The proton would remain on here. 3.3 is more than 1 unit below this guy. The proton would remain on. Same thing here. However, here here we have the pH more than 1 unit below the pKa values, so the proton remains on. But here we see the pH value is more than 1 unit above the pKa. And when the pH is more than 1 unit, 1 or more units above the pKa, we see the proton comes off. And when that happens, we end up with this guy right here. Now we got a minus charge where we had a plus 1 charge. Overall charge of the molecule, plus, 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 minus. Overall charge is equal to plus 2. Okay? Let's go to the other end of the spectrum. Let's say that we had a pH of 13. What's the charge on this molecule? Well, at the other end of the spectrum, the pH is more than 1 unit above the pKa value, so we're going to lose that proton. When we lose the proton, we get NH2, and NH2 has a charge of 0. pH of 13, more than 1 unit above the pKa value there, we get an NH2 again, having lost the proton. We have a charge of 0. Same thing here, NH2, charge of 0. And 13 is more than 1 unit above this. We lose this guy, negative charge, and 13 is more than 1 unit above this, so we have a negative charge here as well. The overall charge of this molecule at pH 13 will be equal to 0 plus 0 plus 0, and that's not a 0, that's a minus, 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 or we have an overall charge of -2. So this simple rule that I've shown you here to take a polypeptide, remember that you've got an alpha amine, an alpha carboxyl, and then R groups to consider. If you start it and you work it sequentially like I've shown you here, you should always be able to predict the charge based on the pH. If you remember the rule, that if the pH is 1 or more units below the pKa value, the proton will be on. If the pH is 1 or more units above the pKa value, the proton will be off. When the proton is on an amine, it has a plus charge. When the proton is off an amine, it has a 0 charge. When the proton is on a carboxyl, it has a 0. And when the proton is off of a carboxyl, it has a charge of -1.